Bismillahirrahmanirrahim, and today we start a new paper which is paper 4.1, and this is the last exam, November 2024, and we will discuss this paper in detail. Bismillahirrahmanirrahim, as you all know, this paper is for 2 hours and it's a very challenging paper, and you must read the questions very carefully, read the whole question and then start answering it, because sometimes the answer you've written in part A could be the answer for part B and you've written it wrong. So just read the whole question and then start the paper. Starting with question number 1, different species of animal have neurons with different characteristics. Figure 1.1 is a diagram of a motor neuron of a rat and a motor neuron of a snail. Now you look at this very carefully. One is the motor neuron of the snail and the one is the motor neuron of the rat. So name the structures labeled A, B and C. Now that's a very simple. A will be the dendrites, B will be the nucleus, and C will be the synaptic knob. So simple A, B, C, A dendrite, B nuclei, because it's pointing to two nuclei, so nuclei, and C is the synaptic knob. 3 marks, and you got all these right, you got 3 marks. The rat motor neuron has an impulse transmission speed of 50 meters per second. The snail motor neuron has an impulse transmission of 8 meters per second. Explain why the rat motor neuron has a faster impulse transmission speed than the snail motor neuron. Now, simply, rat has myelin sheath, and so there's going to be saltatory conduction, which means what? Either you wrote that or you wrote the explanation for it, which is the impulse jumps from node to node. So the fact that myelin speeds up transmission, that is a part of the syllabus, which is taught to you by the teacher. If you follow the syllabus very rigidly, myelinated neurons, why they're fast is because the myelin sheath results in the impulse jumping from node to node. Then coming to the B part of the question, figure 1.2 shows an action potential in a rat neuron, and figure 1.3 shows us in the snail. So this is the snail one and this is the rat one. Be very careful that you're reading the right one. Contrast the two action potentials. Contrast. It's not compare and contrast. Compare is similarities and differences. Now it's saying only contrast. So you can see there is greater depolarization. From minus 40, this goes up to plus 20. That from minus 40 goes up to more than plus 40. So greater depolarization. Then action potential slower, takes longer time. Action potential slower in the snail. Of course, we're talking of the snail, you see. Either you talk of the snail or the rat, is up to you, but then you better specify what you're talking about. So if I'm talking about the snail, so that I'm talking about figure 1.3. So figure 1.3, you can see there's greater depolarization. It's from here to here. Here it is from here to here. Then you can see this increase. Then you can see this. So it says action potential slower or takes longer or takes more time, or you can say longer time to return to the resting potential. Now this is returning to the resting potential after this time. This is returning to the resting potential after this time. Minus 40 is the resting potential. Longer refractory period. Refractory period was the time in which it did not respond to another stimulus. And longer hyperpolarization. This hyperpolarization is this part. This is going down here. And this is longer hyperpolarization. And this is returning here very quickly. So hyperpolarization is this. So longer hyperpolarization in the snail. So you can see all these points. So we must give it, which one are you talking about? Are you talking about the rat or the snail? So you write that on the top as a heading. And then you talk about that is snail has a greater depolarization. In this way, you don't have to even give a long comparative statement. And this is happening in the rat and this is, so even you use the word greater, longer, shorter, anything like that, that's a comparative word which you must use, and you must learn to use that in your paper 4s. So greater depolarization, action potential takes more time, longer refractory period, and longer hyperpolarization. Now, just quick revision of it. What is depolarization? And what is repolarization? Depolarization is this thing when the sodium channels are open. And that's why if you have looked at my videos, the Nike, this is the mnemonic. Sodium channels open and sodium ions diffuse in. Repolarization is Ke, potassium exit. Closure of sodium and opening of potassium, and potassium remains after reaching the rested level. So hyperpolarization is this part. This is the hyperpolarization part. So depolarization, repolarization, and hyperpolarization. So in depolarization, sodium ions are diffusing in. In repolarization, potassium ions are exiting. Then another thing about the refractory period, there is an absolute refractory period during the depolarization and repolarization. And then there is the relative refractory period during the process of hyperpolarization. So it will difficult to stimulate it. Refractory means it will not respond to a stimulus. Refractory period. Now coming to question number 2, the leaves of many plants have stomata that show a regular daily rhythm of opening and closing over a period of 24 hours. Daily rhythm. Explain why it is important for plants to open and close their stomata in a daily rhythm. Well, open in day or open in the light to take in carbon dioxide for photosynthesis, or you can say take in carbon dioxide for the Calvin cycle, or you can say take in carbon dioxide for the light independent stage. And they close in the dark. Why? Why do they close in the dark? Is to decrease the water loss by transpiration. Otherwise, water loss would, basically the stomata are to allow gas exchange. But as we say, transpiration or the loss of water is an inevitable consequence of gas exchange. It has to open. Stomata open, so water vapor will diffuse out if there is a concentration gradient. So it's an inevitable consequence, means we can't prevent it. You open the door to allow a friend inside and mosquitoes may come in and there's going to be a draft. Maybe there's a wind outside, so the wind air is going to gush in. So you only want to open the door for your friend to come in, but then all this is going to happen. So inevitable consequence of gas exchange. So open in the day or open during daylight, taken in carbon dioxide for photosynthesis, close in the dark to decrease water loss by transpiration. Then coming to part B of the question, figure 2.1 shows the results of an experiment to monitor this rhythm over 3 days and nights for the plant Arabidopsis thaliana. The percentage of open stomata is shown. Each day consisted of 14 hours of light and each night consisted of 10 hours of darkness, black bar. Figure 2.1 shows that the percentage of open stomata increases in the first 7 hours of the experiment. Describe the sequence of changes that occur in the guard cell that leads to the stomata opening. Now that's a very simple question. It's come many times, a very direct question. Hydrogen ions leave the cell using energy or ATP. So it's an active transport of hydrogen ions pumped out. So there's low hydrogen ions inside, so this becomes more negative. Potassium channels open, chloride ion move into the cell, water potential of the cell decreases, water moves into the cell by osmosis, cells become turgid or they swell or they increase in volume, or you can use the word expand as well, and guard cells inner wall thicker, so the cells bend or they curve apart. So this was a very direct question. Either you know this or you don't know this. You can't make it up. Nothing is a very straightforward, direct question. As you can see, this was for 6 marks. So for 6 marks, you had to write quite a lot. And you should have written a lot so that you could get your 6 out of 6. This was a very direct question. Part C, the experiment was repeated with Aethaliana plants that were left in darkness from 14 to 96 hours. The results are shown in figure 2.2. So day and night. So this is the day part. And then we have the night part, which is in the black. So this is all the night part. With reference to figure 2.1, so we're going to refer to this, explain what figure 2.2 shows about the roles of genes and the role of the environment in controlling the rhythm of stomatal opening and closing in Aethaliana. This is only for 2 marks, as you can see. So I always say is if you're struggling with it, just leave it. So basically, in the environment, light needed to keep the cycle normal to achieve some peak percentage of stomatal opening. Genes have a role in the regular rhythm, and the cycle continues in darkness even without light. So this was a very general question, and a general answer was required out of it. Now, this part of the question was challenging. Why was it challenging? Is that during the light, of course, you can see open stomata was nearly 100%. But during the dark time, a certain rhythm was still maintained, even though it was all night. So the genes must have a role as the cycle continues even in darkness. Because if it was just that light and dark was resulting in the stomata opening or closing, then why was it opening and closing? Why was that rhythm maintained even when it was nighttime? It was all in dark. So that is what how you had to address this question and answer it. Question 3. During aerobic respiration, cells respire substrates such as glucose to produce ATP. Some events that occur during aerobic respiration are: number 1, the respiratory substance break down into smaller and smaller molecules. These series of reactions are described as catabolism. Coenzymes take part in various reactions. In some reactions, coenzymes are reduced or oxidized. Carbon dioxide is released. Aerobic respiration occurs in 4 successive stages: glycolysis G, link reaction LR, Krebs cycle KC, and oxidative phosphorylation OP. Complete table 3.1 to show which events occur in each stage of aerobic respiration. Use a tick to show that the event does occur or a cross to show that the event does not occur. So catabolism, yeah, it occurs in glycolysis, it occurs in link reaction, it occurs in the Krebs cycle, but it does not occur in oxidative phosphorylation. Then coenzyme is reduced or oxidized. Yes, in all of them. Coenzyme NADH is formed in glycolysis, NADH is formed in link reaction, NADH and FADH is formed in oxidative phosphorylation. NAD it gives up its hydrogen. So it's not reduced, it's oxidized. Then a coenzyme forms a covalent bond with a respiratory intermediate. Now that does not happen in glycolysis. It happens in the link reaction, and then it does not happen in the Krebs cycle, and it does not happen in the oxidative phosphorylation. So it forms the coenzyme, forms a covalent bond with a respiratory intermediate. Carbon dioxide is released, nothing in glycolysis, yes. In the link reaction, yes. In the Krebs cycle, not in the oxidative phosphorylation. You see, explaining this, acetyl-CoA is formed in the link reaction. So coenzyme, acetyl coenzyme A. That is why there's a tick in the link reaction. Part B. A new handheld technological device shows the main type of respiratory substrate being used in the cells of a person. The device consists of a carbon dioxide sensor and airflow meter. The person inhales through the device for a fixed time and then exhales into it. The person inhales through the device for a fixed time and then exhales into it. The device calculates the RQ value to show whether the cells are mainly respiring carbohydrates or lipids. Explain how the device calculates the RQ value and how the shows whether the cells are mainly respiring carbohydrates or lipid. Now you can say it is for 4 marks. So what is RQ? RQ is the ratio of carbon dioxide exhaled to oxygen inhaled. Airflow meter measures oxygen inhaled. Carbon dioxide sensor measures the concentration of carbon dioxide in the exhaled air. Carbohydrates have an RQ of 1, lipids have an RQ of 0.7, proteins have an RQ of 0.9. So this is what you are going to tell you, plus it tells you whether it's aerobic or anaerobic respiration. If it is anything above 1 or 1.9.7 are all aerobic, and that's carbohydrates, fats. You see, it's talking here of the main type of respiratory substrate, and that is only carbohydrates and lipids. Proteins are not respired. They are never respired unless you are in the terminal stage of cancer and then you start respiring the protein. That's why people lose a lot of muscle mass if they are in the last stage of cancer. So now in this question, this was for 4 marks. Lipid has a higher energy value. Explain how the device calculates. Okay. Now, no, that's for the next part of the question. Okay, we talk about part 1. Explain how the device calculates the RQ value. So ratio of carbon dioxide. So when you look at the mark scheme, it's very clear that RQ is ratio of CO2 exhaled to oxygen inhaled. Then the airflow meter will measure the oxygen inhaled. The carbon dioxide sensor will measure the concentration of CO2 exhaled. The carbohydrate RQ is 1. Lipid RQ is 7. So you see, why didn't we talk of proteins? The reason why we didn't talk of proteins, because here it says main type of respiratory substrate. Now, proteins are not respired. No, not respired. Only in the late stages of cancer or terminal cases of cancer, then it is respired. Now coming to the part 2 of the question, state the difference in the relative energy values of carbohydrates and lipids as respiratory substrates and explain the reasons for the difference. Now that's very simple. It's come in many questions, many papers. Lipid has a higher energy value, or you can say carbohydrates is about 15 to 17 kilojoules per gram and lipid is 37 to 40 kilojoules per gram. The another part you could say was more CH bonds in lipids and more reduced NAD or FAD formed, because more hydrogen means more of the coenzyme will be reduced, more NADH and more FADH, and every NADH gives you 3 ATP and every FADH gives you 2 ATP, and this will result in a greater proton gradient, and so more protons will pass through the ATP synthase. So writing it down, lipid has a higher energy value, more CH bonds. C6H12O6, glucose, C6H12. I mean, if you look at fat, it will be C17H34. So more CH bonds, O2, less oxygen. So more CH bonds, more reduced NAD/FAD, greater proton gradient established. So that means more protons will pass through the ATP synthase. So more ATP will be formed. Now coming to question number 4, the leaves of mimosa, whatever plants, are made of a number of structures known as pinnae. The pinna fold when the leaf is touched. This closes the leaf. Now they've shown it to you in the open stage and then they've shown it to you in the closed state when it's touched. Figure 4.1 shows an open leaf of M. pudica before it is touched. Figure 4.2. So 4.1 shows it when it's not touched and figure 4.2 shows when it's touched. This closes the leaf. Figure 4.2 and 4.1, you must be very clear about it. Now, reading the question further, a touch stimulus to an M. pudica leaf causes an action potential to be generated. The action potential results in changes in cells which causes the leaf to close. Figure 4.3 shows the mechanism of M. pudica leaf cells that cause the leaf to close. Action potential after touch stimulus: protons pumped into extensor cell, chloride ions and potassium ions move out of the cells, water moves out of the cells by osmosis, cells become flaccid. Now this is what they are telling you about this. The leaves of M. pudica and the leaves of Venus flytraps move in response to touch stimuli, but the mechanism that cause the responses are different. Describe the differences between the mechanism shown in figure 4.3 and the mechanism that causes the closure of the modified leaves in the Venus flytraps. Now the Venus flytrap is in the syllabus. So in the Venus flytrap, if you remember, you touch two sensory hairs. The protons leave the hinge cells and the calcium ions move into the hinge cells. So the water moves into the hinge cells because it lowers the water potential. And then the hinge cells become turgid, swell, increase in volume or expand. So you have to give me that information which is part of the syllabus, what happens in the Venus flytrap. So as you can see, there are 5 mark scheme points and you have to give me any 3 of these. So touch two sensory hairs, protons leave the hinge cells, calcium ions move into the hinge cells, water moves into the hinge cells become turgid. Now coming to the B part of the question, the rate of photosynthesis decreases by 40% when the leaves of M. pudica close. Explain why the rate of synthesis decreases when the leaves become closed, like the diagram that I've shown you. Basically, is less surface area of the leaf exposed, less light absorbed by the chloroplast. Fewer stomata are exposed, so less carbon dioxide will enter. You see, if a leaf closes up like that, or you can say curls up like that, then the surface area exposed for light absorption is definitely decreased. This is only for 2 marks. So you have to really imagine what the answer could have been. And you write a very befitting answer so that you get your 2 marks. So less surface area, less light absorbed, fewer stomata, less CO2 enters. Part C, plants can carry out cyclic and non-cyclic photophosphorylation during the light-dependent stage of photosynthesis. These processes occur at the grana of the chloroplast. Outline the similarities and differences between cyclic and non-cyclic photophosphorylation. Direct question, everybody must know this. I'm sure you all know this. So we can give the similarities. Photoactivation of chlorophyll occurs in both of them. Electrons move along the ETC. Chemiosmosis, ATP produced. This is the similarities, 4 points. The differences were PS1 and PS2 in non-cyclic and only PS1 in cyclic. Photolysis produced in non-cyclic only. Reduced NADP in non-cyclic only. Electrons return to the same photosystem, that is PS1 in cyclic. So similarities, photoactivation of chlorophyll, electrons move down ETC, chemiosmosis, ATP produced. Differences, PS1 and PS2 in non-cyclic and only PS1 in cyclic. Photolysis in non-cyclic only, reduced NADP in non-cyclic only. Electrons return to PS1 in cyclic. Now, if you talk about the oxygen produced, now that is the same marking scheme point as this. Either you said photolysis or you said oxygen produced in non-cyclic only. So it's not a separate mark scheme point. Photolysis and oxygen produced is one mark scheme point. Remember that. You have to think how many mark schemes point are you going to write in order to get. Now, this was a very long question. You had to get 6 marks for this. So in order to get the 6 marks, you better write the whole story. And you better write a good story, a beautiful story, not BS. We will go on to the rest of the questions in the next video, and we will continue with this. And I hope this has all been very helpful. And thank you very much for subscribing and watching these videos. Thank you very much.